Devlin Moyer, Boston University, comparing performance of module detection methods for identifying aging-related protein modules. Most biological phenomena are controlled not by the action of a single gene or protein, but rather by the coordinated actions of a group of genes or proteins. One particular instance of this problem is the study of healthy aging, where the ApoE protein has been associated with healthy aging and extreme longevity, but many other proteins are also clearly involved. In this work, we compare the results of two common techniques for identifying modules of co-expressed proteins when applied to a proteomics dataset from centenarians and both related and unrelated non-centenarians. The proteomics data consists of serum protein levels for just under 5,000 proteins in about 80 centenarians and 140 non-centenarians. Due to the very small sample sizes, we did not divide the data based on centenarian status before using the module detection methods. The methods we used are weighted gene co-expression network analysis, WGCNA, and independent component analysis, ICA. The two methods take very different approaches to the problem of identifying modules, so we were interested to see how the modules they identified might differ. WGCNA finds the pairwise correlations between all proteins and clusters proteins by those correlations, while ICA attempts to decompose the matrix of protein expression levels in each patient into two matrices, one representing a set of unobserved underlying modules responsible for the protein expression levels, and one representing the degree to which each module affects each individual protein's expression level. The key difference between the two methods is that WGCNA will always assign each protein to only one module, while ICA can assign the same protein to many modules. When we ran WGCNA and ICA on the centenarian data, the results from each method were quite different. WGCNA generally made larger modules than ICA, as shown in Figure 2. A few of the modules identified by WGCNA were somewhat similar to a module identified by ICA, using the Jacquard index as a measure of module similarity, but most modules identified by WGCNA were not particularly similar to any module identified by ICA, see Figure 3. To investigate the possibility that the small sample size was to blame for the differences in the results of the two methods, we constructed a simulated network of co-expressed proteins where the module membership of each protein was known, as shown on the left panel of Figure 4. Using this network structure, we generated 100 expression datasets at several different sample sizes and ran both WGCNA and ICA on each one. The results were somewhat reminiscent of the centenarian modules. Once again, ICA assigned far fewer proteins to modules than WGCNA. Compare the number of white nodes in the right panel of Figure 4 to the number of gray nodes in the middle panel. These represent proteins not assigned to a module. Note the network structure remains the same in all three panels of Figure 4. Only the colors of the nodes differ, indicating the module membership of each protein. Since the true module membership of each protein was known, we could score each module detection method for accuracy. See Figure 1 for a definition of this score. As shown in Figure 5, WGCNA significantly outperformed ICA on every sample size used. It should be noted that neither method scored particularly high, as the score we used ranges between 0 and 1, and even with 2,000 samples, the highest score by either method was only slightly above 0.6. These findings echo a well-known problem with the study of rare phenotypes such as extreme longevity. Small sample sizes significantly impede our ability to comprehend the underlying biological processes, but no large sample sizes exist. Our results also showcase how different the output of these two widely used module detection methods can be, both on real and simulated datasets. It is possible that a new module detection method could be devised to be more capable of identifying modules given small sample sizes. Such a method would be very useful for the study of aging, as well as numerous other rare phenotypes.